In this video, I'll be describing the function of cytotoxic T cells. Cytotoxic T cells, also sometimes called cytotoxic T lymphocytes, are able to recognize cells that are infected with a virus. So when a cell is infected with a virus, it's going to take over and start producing these unusual proteins. So this is a protein that is not normally produced by the cell, but is made for making virus particles. So cytotoxic T lymphocytes can recognize that, and once a cell has been kind of taken over by a virus, you can't really get the virus out. And the most effective way to protect the body is to kill the cells that are infected. So let's look at how cytotoxic T cells are able to recognize these virus-infected cells. So here we have our cell. And all cells actually produce MHC1. So any cell in our body is normally producing proteins. Here's the ribosome, producing some protein through the normal transcription and translation process, producing these proteins here. And MHC1 is very similar to MHC2, but it's going to bind to proteins that are produced inside of the cell. And in a similar way to MHC2, those molecules will get brought to the plasma membrane. And here it is presenting that antigen. So this is a normal antigen. This is a normal protein that's being produced. And the cytotoxic T lymphocytes are going to actually ignore this. They've been trained to know what belongs there and what doesn't. So what happens if we have a cell that gets infected by a virus? So now we have these unusual viral proteins that are being made. And these viral proteins are also going to bind with MHC1. These are all MHC1. And that MHC1 is just kind of always picking up a little bit of what's going on inside the cell. It's kind of an internal surveillance system. So now this MHC1 is presenting a protein that is unusual, and the T cells have been trained to ignore things that are normal. But now our cytotoxic T lymphocyte, or cytotoxic T cell, is able to bind with its T cell receptor. We can just kind of draw that T cell receptor smaller here. That would be bound to that whole T cell. And it now recognizes an unusual protein. Because it's been trained to ignore things that are normal, so anytime it sees something unusual, it's going to kind of trigger that cytotoxic T lymphocyte. And so here we can imagine this virus-infected cell with MHC2, and it's got this antigen on there that's a match for that cytotoxic T lymphocyte. So here's our infected cell. And here's our MHC1. And so this cytotoxic T lymphocyte recognizes that antigen there. And that's going to cause this cytotoxic T lymphocyte to kill that infected cell. So how is it actually going to do that? These cytotoxic T cells release these enzymes called perforin and granzyme. And these enzymes are toxic to all cells of the body. And so they've got this targeted attack specifically aimed at that cell that they're binding to. And this is going to lead to a process called apoptosis, which is a way of killing that infected cell. Apoptosis is also called programmed cell death. Sometimes people also call it cell suicide. So it's triggering this process that leads to the cell killing itself. And this activates a whole bunch of events, including breaking down their own DNA. They'll start to digest their own DNA, and they'll start to break down the cell into little pieces. And so we actually see that kind of starting to happen here. The cell is sort of breaking itself up into little bits. And eventually those little bits will just be kind of broken away from the cell, and the cell will be disintegrated into nothing. And those fragments can be picked up by macrophages or other parts of the immune system. So we've talked about helper T cells and cytotoxic T cells. So why don't we go ahead and do a quick review of each of these type of T cells. So as a reminder, all T cells bind specific antigen in their T cell receptor. So there has to be that specific match. But they can't see the antigen on its own. They have to bind that antigen with MHC. Helper T cells are going to recognize antigen that comes from a phagocytosed pathogen. Or something that was eaten by an antigen-presenting cell. Whereas cytotoxic T cells are going to recognize internally produced antigen. And by that I mean inside of the cell. So these are usually viral proteins. Things that are actually produced by that cell itself. Helper T cells are going to recognize that antigen that's presented on MHC2 and cytotoxic T cells are going to recognize antigen presented on MHC1. And when this helper T cell recognizes that antigen presented by the MHC2, it's going to lead to activation of the T cell, and the main things that are really going to help fight the infection are going to be further activation of B cells. They're going to activate macrophages. And also, we didn't talk about the details of this, but they are needed for activation of cytotoxic T lymphocytes, or cytotoxic T cells. So these helper T cells, in a way, are kind of the regulators of the immune system. They're overseeing all these different functions of the immune system. And the cytotoxic T cells, their main function is to kill infected cells. So they're both recognizing unique antigens, but the end result of what they do is quite different.